The five kingdom system of Robert H. Whittaker, although a major improvement over the simple two kingdom system of plants and animals, still left much room for criticism. While the kingdom Monera, with its distinct prokaryote organization of the nucleus, is still considered the most natural and ambiguous kingdom of this system, the remaining kingdom have all undergone some or other taxonomic scrutiny. The plant, animal and fungi kingdoms, all multicellular eukaryotes, have each been defined by their nutritional modes. Several researchers have criticized the use of these characters, arguing that they define adaptive traits and not taxa. These names are thus entirely unsuitable for revealing phylogenetic relationship among organisms. The kingdom Protista or Protocrista is the hardest to define, as it is the least homogeneous group. It is somewhat of a mix and match containing all eukaryotes that do not fit into the plant, animal or fungal kingdom. Now, let us see some points towards the 8th and 6th kingdom systems. The problems encountered with the 5 kingdom system saw the continued use of the plant, animal and fungi kingdoms. The former kingdom Monera, the least problematic kingdom, was split into the kingdoms Eubacteria, that means true or present day bacteria, including the cyanobacteria, and the archaeobacteria, that means ancient bacteria. Despite being readily accepted, many still have problems with the placement of the cyanobacteria or blue-green algae in a bacterial kingdom. Time saw the erection of the new kingdom Chromista, proposed to include the helps and other brown algae, that means phaeophytes, the diatoms, that means basilarophytes, the golden brown algae, that means chrysophytes, certain molds, that means oomycetes, and even heterotrophic flagellates, that means silicoflagellate. At this point, the 8th kingdom system of classification was born. Advances in molecular phylogeny, however, saw the development of a 6th kingdom system developed by Cavalier Smith in the year 1993, with the proposal for the erection of the kingdom Protozoa separate from the kingdom Chromista. The kingdom Protozoa occupying a pivotal position between the ancestral prokaryotic kingdom bacteria and the 4 derived eukaryotic kingdoms Animalia, Plantae, fungi and Chromista, that was developed by Cavalier Smith in the year 1998. The Chromists represent an independent evolutionary lineage that appears to have diverged from the same common ancestor as plants, animals and fungi. The suggestion of a divergent algal group, however, being placed in a newly erected kingdom, was met with much criticism because the precise relationship of the chromists to the other eukaryote is still problematic. However, the Chromista as a group was registered and named under the International Code of Botanical Nomenclature that was developed by Golding in the year 1996. The name Chromista means colored, and although some chromists like the Oomycota, Saginista and various chrysophytes and silicoflagellates are colorless, the majority are photosynthetic. Despite their photosynthetic capacity, chromists are not all closely related to plants or even other algae. Unlike plants and green algae that possesses chlorophyll B, in addition to chlorophyll A, the Chromista possess chlorophyll A and C. Though the reserved food is starch, it is observed that the main food reserves of the Chromista are the oil leucine and the polysaccharide cresol aminorin. Beta 1,3 linked glucan that is formed in vesicle located outside the chloroplast. Photosynthetic chromists often also carry various accessory pigments that mean fucoxanthin in some, Vaccaria xanthan in others, that give them their characteristic golden to brown to orange coloration. But it is not their photosynthetic and accessory pigments that characterize the Chromista, but rather an unusual suite of diagnostic feature found in no other group. The group originally got its name from the fact they all possess flagellated cells at some stage of their life cycle, and the flagella are typically of two types, subsequently referred to as the heterocon type flagella. One type of flagellum is covered with thin, stiff tubular hairs and is usually directed into the direction of swimming. The other type is shorter, lacks a coating of stiff hairs and is usually directed away from the direction of motion. The anterior flagellum is referred to as a tin cell. Pleuronematic or flimmer flagellum, while the thin hairs are called mastigonemes. Whenever present, the mastigonemes have a unique tripartite structure. The posterior flagellum is referred to as a whiplash flagellum. Secondly, their chloroplast and nuclei are inside the rough endoplasmic reticulum, that means ER. Instead of free in the cytoplasm, that is, the chloroplast is founded by a double membrane, but has an extra layer of endoplasmic reticulum, that means also two layer, that is often continuous with the nuclear envelope. Inside the chloroplast, the thylakoids are staked in groups of three called lamella. The chloroplast contains a light sensitive eye spot of pigmented granules called the stigma. The chloroplast DNA is not bounded by a three membrane and usually arranged in the shape of ring called nucleoid, that means nucleomorph or cryptonucleus, and is thus a degenerate nucleus. The nucleoid is a simple single stranded DNA molecule coiled around an RNA core and has also been known as bacterial chromosome or a chromatin body. A third character common to the Chromista is their mitochondrial architecture. Mitochondria are generally spherical organelles that have an outer membrane surrounding an inner membrane that fold, that is called cresta, into scaffolding for oxidative photophosphorylation and electron transport from enzymes. While most mitochondria have flat, self-like or flight-like cresta, the chromists have tubular cresta. Majority of those that do not possess silica or calcium carbonate skeleton deposit various cellulosic compounds around their cells. This gives them form and rigidity while remaining will be flexible. Now we will see some point to study the characteristic of chromist and to study the detail of morphological characteristic of the members of chromist. How did the chromist arise? Let us discuss. It is now generally accepted that present day chromist arose from a single endosymbiotic merging event between two eukaryotes into a single cell. This through the process of secondary endosymbiosis. It has been postulated to have occurred during the latest stage of the symbiotic origin of mitochondria and chloroplast. The evidence is based largely on the presence of unusual organelle structure such as the nucleoid, that means nucleomorph or cryptonucleus, a naked, that means membrane free structure containing DNA in the shape of a ring located within the endoplasmic reticulum. This minute well hidden structure codes for 80 ribosomes that are distinct from those of the cytoplasm and is inferred to be reduced nucleus of the former endosymbiont. The second line of evidence involves the double membrane of the endoplasmic reticulum surrounding the chloroplast. The two membrane of the chloroplast endoplasmic reticulum are very different in evolutionary origin and must therefore have very different physiological and developmental properties. The outer rough membrane belongs to the host, while the smooth inner membrane is in fact the linear descendant of the plasma membrane of the eukaryotic symbiont that provided the chromist chloroplast. Even today, this smooth inner membrane seems to undergo budding to genera. Now, we will see some common character to the taxa of the Chromista. Regarding main pigments, Chromista containing chlorophyll A and chlorophyll C, alpha and beta carotenes, xanthophylls, that mean vicosanthan in some, Vachuria xanthan in others. Chlorophyll B is never present in Chromista. Regarding food reserves in Chromista, crystalaminorin, that means beta 1,3 linked glucan, formed in vesicles outside the chloroplast. Regarding the chloroplast feature, Chromista containing membrane 2-layered but with an extra 2 layer of endoplasmic reticulum that is continuous with the nuclear envelope. The thylakoids are staked in groups of 3 lamella. Regarding chloroplast endoplasmic reticulum, this is present in Chromista. Regarding cell wall of Chromista, it is varies, sometimes includes silicon. Regarding the flagella, in at least some stage of its life cycle, one flimmer or tinsel and one smooth, that is whiplash, collectively referred to as the heterocon type of flagella. Mitochondrial cristae, in Chromista it is in tubular form. Now, we will study the different group of chromists. First, we will see the silicoflagellata. The silicoflagellata are uncommon, mostly planktonic, marine, flagellated algae that are represented by both photosynthetic and heterotrophic forms. They are characterized by having internal skeletons, that means distinctly star shaped, over which the cell body wraps itself. The skeletons are constructed by a secretion of silicon dioxide either in the form of a network or framework, that means resembling simple radiolarians, or in the form of multiple scales. They possess the typical heterocon type flagella. The long flagellum called an undulipodium is used to propel the cell, while the other flagellum is much reduced and barely visible. They reproduction by simple mitotic division. Many others classify the silicoflagellate as a class under the Chrysophyta, that means, and it is also referred to as the Dictyocopaceae. Others have, however, suggested the elevation of this group, arguing that the Chrysophyta are almost exclusively freshwater algae, while the silicoflagellate are almost exclusively marine. The next one is Chrysophyta. Although the Chrysophyta, that means golden or golden brown algae are now generally believed to be paraphyletic or possibly even polyphyletic. That was referred by Zangrin and other co-workers in the year 1995. Many still treat the subtaxa belonging to this group as a composite entity. Their distinct golden or golden brown coloration is derived from their yellow, brown and orange accessory pigments. That means carotenoids and xanthophylls. Fucoxanthin is particularly important in denoting color to this group. Although it is entirely absent from the Xanthophyceae, they are apparently unifying characters are the presence of chlorophyll A and C and the possession of the heterocon type flagella. There is, however, a small minority of chrysophytes that are colorless and are essential heterotrophic. Food is generally stored as oil or as the polysaccharide lamellin. That is also very typical of the brown algae. Because of the heterogeneous nature of the Chrysophyta as a group, several subtexts are formally included in the Chrysophyta have been given separate recognition. Eustichometaphyceae, also referred to as Eumasticophyceae, Formophyceae, Rabidophyceae, also referred to as the Rabidophyta. Chloromonads, Sarginochrysophyceae, also referred to as the Sarginochrysophyceae. Next, we will see the Vigera or Oomycota. The Oomycota are quite literally egg fungi, also referred as pseudofungi. Is a small division of fungal-like filamentous proteus commonly known as water mold and downy or powdery mildews. Although some are unicellular, the majority are multicellular and mycelial. That means branched filamentous cenocyte in nature. The Oomycota have been classified as Chromist because their free-swimming juice pores possess the heterocon type flagella. Food is stored in the form of mycolaminarin, an energy storage molecule similar to that found in diatoms and brown algae. For some time, however, the oomycetes were classified as fungi because of their essentially filamentous growth and feeding mode similar to that of the fungi. Unlike fungi, however, that have cell walls composed of chitin. The oomycetes cell walls are composed of cellulose similar to that found in the brown algae. Also, the free living stage of the oomycete has a diploid chromosome complement while that of the fungi is haploid. Next, we will see something about Heptophyta. Heptophyta, also called the Primnicophyta or Coccolithoporids. Heptophyta with the Chromista despite growing evidence to the contrary. Typically unicellular flagellate, the heptophytes were once classified with the Chromist, that means heterocon. On the basis of their photosynthetic pigments, that means more specifically the accessory pigments diadinoxanthin and fucoxanthin, and together they were classified as the Chromophyta. This was particularly in reference to their golden coloration. The Heptophyta, however, lack several of the morphological and molecular characters common to the Chromista. They contribute to atmospheric dimethyl sulfide, which seeds clouds and forms rain, including acid rain. Fucocystis poucheri can occasionally form foamy mucilaginous bloom that suffocate marine life. A native species of Primnicium has also been implicated in fish kills. Other species can cause non-toxic bloom, and the skeletal remains of some species in marine sediments are useful paleoindicators of climatic change. Next, we will see Heliosova, that means sun proteus used to be classified among protozoans, but molecular evidence suggests they are chromist, most closely related to heptophytes. Only 3 species have been formally recorded, even though Heliozoan are known to be common inhabitants of freshwater and marine environment. Next, we will see Saginista. Saginista are like the haptophytes. It is well established that the Saginista is a basal group of protists. While there is much debate about the phylogeny of this group, there appears to be consensus as to the paraphyletic origin of this group. Although the specific relationships are unclear, the Saginista is generally divided into two groups, namely the Bicocca. This is also referred to as the Bicocca flagellata or Bicoccida, commonly referred to as the Bicoccocids or Bicoccid, and the Labyrinthulomycota or Labyrinthulia. The Labyrinthulids, the photosynthetic and heterotrophic unicells. Although poorly studied, it is known that photosynthetic unicells possess chlorophyll A and chlorophyll C and the typical heterocon type flagella and the tubular cresta in their mitochondria, features that they share with the majority of the chromists developed by Gavalier Smith in the year 1989, believes the bicoccid to represent the primitive protist that first entered into the symbiosis with a chlorophyll C containing symbiont to form the chlorophytes. Like the bicoccocids, the labyrinthulids, more commonly known as slime nets, also have flagellated stages in their life cycle that possess the typical heterocon type flagella. Although they have traditionally been classified with the fungi, it is their flagella feature that group them with the chromists. Their common name derived from the fact that they are unique in their ability to produce or secrete an anastomosic slime net or membrane outside their cells. Hardly any characters unite the bicoccid and the labyrinthulids, and many others have opted to separate these two groups. Next, we will see something about Basilauriophyta, commonly known as diatoms. The Basilauriophytes are unicellular or colonial, but are never organized into more complex thalli. Only the male gamete are flagellated with the flagella typically of the heterocon type. Like most chromists, they also possess chlorophyll A and chlorophyll C. The chief distinguishing feature of the Basilauriophyta, however, is the cell wall, which is siliceous and composed of two overlapping halves, rather similar to Petidis. This type of cell is termed frustule. The two kinds of pinnate and centric diatoms are classified into two orders, the pinnales and centrales. The frustules of pinnate diatoms are usually elongate and bilaterally symmetrical in wall view. There is usually an elongate structure, that means the raphe, visible at the surface of each wall. This is actually a slit in the frustule, at the center of which is a thickening called the central nodules. At each end of the raphe, there are also thickening called polar nodules. Mucilage extrude through the raphe or polar regions may be used for movement, attachment or protection from abrasion. There are many features present on the surface of the frustule, many of which are best resolved using the scanning electron microscope. It is this pattern that means marginal projection and minute holes called punctae. They are used to separate taxa with the Basilauriophyta. Next, we will see about the Xanthophyta. The Xanthophyta, that means variously also referred to as the Xanthophyceae, Tripophyceae and the Heteroconta are commonly called yellow-green algae. Unlike the other chromists, the Xanthophytes lack the accessory pigment fucoxanthin, the pigment responsible for the golden brown to orange coloration of its sister taxa. The dominant pigment in the Xanthophyta is chlorophyll A, which generally gives the algae a green coloration. This combined with the fact that members of the group often produce copious amount of carotenoids gives the xanthophyte their distinctly yellow-green coloration. Those xanthophytes that have reduced carotenoid concentration are quite easily mistaken for green algae. A suite of distinct features characterizes the xanthophyta. They are primarily freshwater and soil algae. That means often also occurring on and within the plant surfaces, but few planktonic marine forms, although most occur as either flagellated unicells, coccoid or filamentous forms. Some occur as palminoid colonies, while other most distinct forms are based on a cenocytic or siphonous organization. Like all chromists, they possess chlorophyll A and chlorophyll C in only small amounts. The zooids have 9 typical catacon type flagella, although these are apically inserted, and food reserves stored as chrysolaminarin in many xanthophytes, like so many of the other taxa, is by no means adequate. The xanthophytes typically lack the nucleoid present in most chromists, suggesting a secondary acquisition of the chloroplast. While there is still much debate as to the placement of the xanthophyta, the group as a whole has even been suggested to require revision at the generic, familial and ordinal levels. This is in view of the fact that molecular evidence now suggests that the character used to separate the xanthophyta at the ordinal level appears to no longer be valid. Next one, Phaeophyta. The Phaeophyta, also referred to as the Bacopaceae and on occasion the Chromophyta and the Chromophycota or the brown algae. The Phaeophyta, being the largest of the chromistid, only occur as multicellular forms. They range from filamentous species, for example we can quote Ectocarpus, to complex parenchymatous species reaching up to 60 meter or more in length. X amplified by the giant helps, that mean macrocysts of the Californian coast. There are between 1500 to 2000 species known at present, assigned to approximately 265 genera. Only 6 genera are represented in freshwater, the rest being entirely marine. The Phaeophyta are undoubtedly members of the Chromista, sharing many of the characters that distinguish the group. They possess chlorophyll A and chlorophyll C, lack of chlorophyll B, and possess the accessory pigment fucoxanthin. The products of photosynthesis are stored as laminarin. Motile stages are generally pear-shaped, that means fiery-form, and have the typical catacon type flagella, namely two laterally inserted flagella, one of which points forward and the other backward. Food is stored in a stalk pynoid that is attached to the chloroplast. The cell wall comprises fluorotenins and is made up of 10 cellulose strengthened by calcium alginate, a substance that is of considerable economic significance as it is extracted and used as a gelling and emulsifying agent in a large number of industries. While the placement of the Phaeophyta within the Chromista is well established, the exact relationship of this group to other chromists is still vague. Many believe that they are most closely related to the xanthophyta and then secondarily to the chrysophyta. Now let us see some summaries. In this topic, we are covering the important characteristic of Chromista as they do not find position in the 5 kingdom classification, and very less information is available on this species. The members of Chromista are single endosymbiotic, merging even between two eukaryotes into single cell. This through the process of secondary endosymbiosis. By the study, many literatures suggested that name Chromista means colored, and although some chromists like the Omycota, Saginista and various chrysophytes and silicoflagellate are colorless, the majority are photosynthetic. The chromists represent an independent evolutionary lineage that appears to have diverged from the same common ancestor as plants, animals and fungi. The suggestion of a divergent algal group, however, being placed in a newly erected kingdom was met with much criticism because the precise relationship of the Chromist to the other eukaryote is still problematic. However, the Chromista as a group was registered and named under the International Code of Botanical Nomenclature.